This PowerPoint will cover the gram-negative Diplococci and the Coccobacilli. Neisseria species are recovered from the human respiratory and urogenital tract, and these organisms are transmitted from person-to-person contact. The normal pathogens, or the most predominant pathogens that are found, are the Neisseria gonorrhoeae and Neisseria meningitidis. These organisms are all aerobic organisms. They are gram-negative Diplococci, and they all are oxidase positive and catalase positive. Neisseria gonorrhoeae is the second most commonly reported sexually transmitted infection within the United States. And this causes the infection that we know as gonorrhea. This infection is typically localized to the mucosal surfaces, and with males they usually exhibit uritis with dysuria and urethral discharge, and with females this organism will colonize the endocervix and will cause the vaginal discharge, dysuria, and abdominal pain. And this organism can also infect other sites like the eyes, the throat, the rectum. With these infections, if they are left untreated, then they can disseminate, which can lead to things like the pelvic inflammatory disease or purulent arthritis and septicemia. And because this is transmitted vaginally to the infants, if the mother does have the organism, then the babies are all given a erythromycin optic solution that once the baby's born, they'll put the solution over the eyes, and this helps to prevent any sort of trauma to the infant's eyes from this infection. And if you've ever had a baby or seen any images or pictures of babies right after they're born, you can see where they have that little gel type solution over the infant's eyes. The virulence factor includes the surface villi, which it helps this particular organism to actually transfer genetic material between different strands of the organism. It allows it to attach to the mucosal surfaces, and it also allows it to invade the host. This organism is also able to survive any sort of attack response from the host because it is able to prevent the cells, the neutrophils, from actually phagocytizing this particular bacteria. And for that reason, you can actually see it inside of the white blood cells if you were to do a special stain to look at those. It also has a capsule. It produces an endotoxin, and then the outer cell membrane proteins, they are responsible for eliciting the different inflammatory responses within the host. And then the outer membrane proteins are involved in the attachment and invasion into the actual mucosal surfaces. So when you're working up Neisseria gonorrhoeae, this particular organism will typically not grow on blood agar, but it will grow on chocolate agar. So with this organism, what you're looking for on the chocolate agar is small grayish-white, convex, translucent, shiny type colonies, and they can have smooth edges or slightly irregular edges. This organism is an aerobic organism, so it can grow in the presence of oxygen. And of course it gives a gram-negative Diplococci stain when it's gram stain. They are oxidase, catalase positive, and DNase negative. And if the main test that you do to work up these organisms is the carbohydrate utilization test. And so you could do this in a test tube, but here at the bottom there's an image of some more newly developed test methods, and there are several that look similar to this, or they may vary to a certain extent, but inside each of these wells you have the different carbohydrate solutions. And so for this one here, C is your control, and then you have your glucose, maltose, lactose, and sucrose. So you have your sugars that follow. Then you have a DNase test and a beta-lactamase test. But what you're looking for is to see if this organism is able to utilize these sugars. So you're looking for that yellow color change where it has utilized it. And so this particular organism is able to utilize glucose. So your control would be here, which would be a negative. You wouldn't expect any sort of change. If this did change, then your results to follow wouldn't be valid. And so with this particular organism, because it does utilize the glucose, but negative for everything else, so these are all negative, then that gives you your results here, where it's able to utilize glucose but nothing else, and that's how you would differentiate Neisseria gonorrhoeae. Neisseria meningitidis is transmitted through respiratory droplets. It can also be transmitted, it's suspected to be transmitted through sharing of drinks and things like that. It mostly affects the adolescent crowd, and this is usually seen with like college dormitories, military dorms. However, vaccination over the years has helped to control and prevent such a severe form of the disease. It can cause fatal bacterial meningitis in children and adults, and so this is usually accompanied also with a type of rash, a petechia type rash, and it is also associated with bacteremia. And so what this can do is lead to things like thrombocytopenia, DIC, and shock, with the DIC, disseminated intravascular coagulation. So this is where the coagulation is actually affected, and you have clots happening in areas of the body where it shouldn't happen. And then you also have other areas that are bleeding out from where those clotting factors are disrupted. It can also lead to the patient actually bleeding out in death if a patient experiences this. So with this particular organism, it is the leading cause of community-acquired meningitis. Other sites that can be affected would include conjunctivitis, sinusitis, and different infections throughout the organs, and a pneumonia without the actual spinal meningitis, bacterial meningitis. So with this organism, once the patient comes in contact with it, it first, has to use its virulence factors, where it has the surface villi that allow it to attach to the mucosal membranes and actually start invading through those epithelial cells and through the submucosal membrane layers. And then it has the capsule that helps to prevent it from being recognized or phagocytized by the neutrophils. And then once it has survived and is able to grow and spread throughout the bloodstream, then it can start producing a more systemic type infection, and which could also include the release of the endotoxins that can create something like a shock. And then you have the IgA protease that increases the host susceptibility to the invasion, so it actually allows this organism to be a little bit more invasive. And so you can perform different serotyping to help identify this organism, and there are commercial kits out there that help with this. And so when it would be useful is if you're looking for, if you have an outbreak within a college and you're trying to kind of link this outbreak to the same organism, and this is where you would actually do the serotyping. When working up this organism, it can grow on blood agar, but it's not as likely to see it growing on blood agar. So again, this is something that would be cultured on a chocolate agar. So this is where the laboratory has to know the source, so it's very important when submitting samples that the source is reported. Of course, with this particular infection, it would be a spinal fluid, so there would be various tests that would be ran on the spinal fluid, including looking at a gram stain where you're looking for the gram-negative Diplococci, and it would be plated on chocolate agar because you would be expecting that this could be a potential cause of the infection if you were expecting meningitis. And so when it grows on chocolate agar, it will be medium to smooth round colonies that are moist, gray to white. And you can have some encapsulated strands, so they may have a mucoid type look to them. And it can give a greenish cast to the agar underneath those colonies. And you can see that a little bit here in this image where it kind of has a tinge to it in that 48-hour growth. These organisms are aerobic, and you can see them intra and extracellular, so you can see them actually inside of the white blood cells. So if you were looking at a gram stain, this isn't a very good image of it, so this would be a cell here, and you can see it looks like it's probably one there. And these are probably also intracellular. You cannot see the outline of that cell, but they do look like they're around the nucleoli or the nucleus here. And so you can see those gram-negative diplococci there. These organisms are oxidase positive, catalase positive, and DNase negative. And again, when you're looking at carbohydrate utilization, these organisms use glucose and maltose. You can see that glucose and maltose there at the bottom have turned yellow. Moraxella catarrhalis. This particular organism is normal microbiota of the upper respiratory tract, and it can occasionally colonize the female genitalia tract. So these organisms are spread because of the patient's normal strand. So it can go from where it would normally be found to a site that would usually be considered a sterile site. And so this is a person-to-person spread through the respiratory droplets. And this particular organism will cause a respiratory tract infection, and it's most commonly localized to sites that are associated with the respiratory tract. So this would be like your ears, sinuses, and it can cause pneumonia. And so a lower respiratory tract infection is often seen more commonly in your elderly patients or those with some sort of like chronic obstruction pulmonary disease, so your COPD. And so with patients elderly patients or COPD patients that present with a pneumonia type infection, then this would be one of those organisms that would be suspected. So this organism grows on chocolate agar with a pinkish brownish opaque type hue to it. These organisms they demonstrate something that's very unique, which they have a hockey puck type a demonstration. So if you would actually use a loop to push this colony, it would actually slide across the agar and not break up. It's not sticky or anything. So that is one of the characteristic things that if a tech is working this up and it looked suspicious to be Moraxella, then they would actually see if they could push that colony across the plate. These organisms are aerobic. They are gram-negative diplococci, oxidase positive, catalase positive, and DNase positive. And these organisms do not utilize any of the carbohydrates, so they would be negative for glucose, maltose, lactose, and sucrose. Haemophilus species are small pleomorphic gram-negative Coccobacilli. They are non-motile and oxidase positive. They are aerobic or facultatively anaerobic, and they require growth factors that can be found in blood, which include the X factor or the human factor, or V factor, which is the NAD. And so these particular factors are found in chocolate agar. Sheep blood agar only has factor X, so the only way they would grow on the sheep blood agar would be if they were growing around a Staphylococcus aureus, and that's because that the staph does produce the V factor, and so because of that production, then the organism could get the X from the actual sheep blood agar, and the V factor that's produced by the Staphylococcus, so they would grow around that Staphylococcus. Our common pathogens are Haemophilus influenza, and Haemophilus ducreyi. Haemophilus influenza it has two categories that are based on the capsule characteristics. So you have typable strands that produce the capsules, and there are 6 groups of that are groups A through F. The group B, or type B, Haemophilus influenza, is one of the most commonly encountered serious infections of this particular organism, and this is where we get the Hib V vaccine, so the Haemophilus influenza B vaccine. There are also non-typable strands, and these strands don't produce a capsule, and they are most often associated as the normal upper respiratory tract flora. Then you also have a biogroup of the Haemophilus influenza that can produce infections as well. So you will occasionally see this particular biogroup in culture. Haemophilus influenza is normal upper respiratory tract microbiota. It causes infections, especially in children, and so this is spread through droplets or through contact. It can cause meningitis, septicemia, pneumonia, and so these infections can be due to the encapsulated strand, or it could also be it's from encapsulated strands, so it can cause something as severe as the meningitis with encapsulated strands, and then something like conjunctivitis or ear infections with the unencapsulated strands. So these are gram-negative Coccobacilli. They're non-modal, catalase positive, oxidase positive. And so when these organisms are cultured on blood agar, they will not grow unless there is Staph aureus present, because these organisms do require X and V factors. And so because they require X and V, then it needs to be a media that provides both of those, so the chocolate agar or a blood agar that actually has the Staph aureus on it. So if you look over here to the right, then you can see where this particular organism is doing what we call satellitism or satellite growth, where you actually have the staph streaked down the middle. If I can get my pointer right. So you have staph streaked down the middle. So there's a culture streak of Staph aureus. And then you can see the growth here around the edges where you have all those small pinpoint colonies that are very translucent, that are growing because of that V factor that is produced from that staph. And then here up top you have where there's an infant that has septicemia from this organism, and then it has transitioned to like a DIC where you have this disseminated intravascular coagulation. So you can see where there's some necrosis of that skin tissue on that infant. Haemophilus ducreyi, or Haemophilus ducreyi, depending on who pronounces it. This organism is transmitted sexually through the genital tract, and so this is not normal flora. And what it does is it causes genital lesions, and so you can see down here at the bottom, of course these aren't images of genital lesions because I'm trying to keep it a little PG rated here, but these are similar to the lesions that it would cause, those open wound type lesions. And so this organism is small pleomorphic gram-negative Coccobacilli or rods. And what you see with this one on a gram stain is a characteristic school of fish type appearance, where they kind of almost in clusters, but where these organisms are really close together, almost like you'd see like a school of fish. So you see all of those like pleomorphic type rods. So you can see this one here, you've kind of got 2 of them together where they're the Coccobacilli, they're kind of elongated out a little bit. And so these organisms are non-modal, catalase negative, oxidase positive. They will grow on the blood agar plate because they do require the X factor, not the V. So not only will they grow on blood agar because it has X, not V, but chocolate agar as well because it has X and V. These organisms will produce a gray, yellow, or tannish type non-mucoid colony. But in order to identify this organism, most of these are going to be worked up through some sort of nucleic amplification, so some sort of STD probe where they'll actually do automated testing on these. So they would look for this particular organism based on that DNA type probe. Brucella species are categorized as a Category B select agent. So these organisms are handled with caution in a biological safety hood, and they would be reported. And so this is a zoonotic disease, so they are transmitted through animals, and they do tend to localize in tissue that's rich in a particular sugar compound. And so these organisms are transmitted either through ingestion of infected unpasteurized animal milk, so like goat milk, through inhalation of the particles that have been aerosoled, through direct contact of infected animal parts, and through accidental inoculation of the mucous membrane, so introducing it to the mucous membranes. These organisms are actually ingested through the neutrophils, and once they're in there, they replicate inside those neutrophils, and then they are released from those neutrophils, which could be through destruction of the neutrophil itself. And so if these infections are left untreated, then they can cause granulomas in different organs, and they can also cause hepatosplenomegaly. This organism is identified through based on its characteristics of being a gram-negative Coccobacilla or short bacilli. And so they stain very poorly on a gram stain, and they can be intracellular organisms, so you can see it inside of those neutrophils because, again, it uses those neutrophils to replicate. They are aerobic cultures, although you do have some that prefer the CO2 environment for growth. They will grow on a blood agar and chocolate agar, but they do require an extended incubation. They produce small colonies that are convex, smooth, translucent, and they can be they're non-hemolytic, and they can also have a slightly yellow tinge to them. They do not grow on McConkey, and so these organisms do better if they're actually cultured on a Brucella type agar that's specifically for these organisms, so those agars may be used if it is suspected. They are oxidase positive and catalase positive, and so usually these organisms are going to be detected through some sort of automation or PCR rather than its biochemical traits that you would see with some of the other organisms that we've previously talked about, where you work those up with different spot tests. Bordetella pertussis. This organism will enter the respiratory tract once it's inhaled, and then it binds to and destroys the ciliated epithelial cells that line the trachea and the bronchial tract. And so with these organisms, they have several virulence factors, one of which is adhesion. And so with this one, it will bind and actually start to paralyze the cilia, and then it will produce a tracheal cytotoxin. And then it also produces the pertussis toxin along with several other toxins. And so these toxins will actually enter the target cells, and it will actually activate the paralysis or the destruction of the different cells. And then it also has the ability to overcome the host defense, and this is because of an outer membrane that it produces. So this particular organism, it mostly affects, it can affect different ages, but we usually do see this more or associate more with children, and what we call whooping cough. So with this, it starts out with very mild symptoms, maybe cold-like symptoms, and then it will progress to a severe violent cough, and this is where you get that whooping cough, where it has that whenever the child, once they cough and they're trying to breathe back in. And then it can go into a convalescent stage where it actually affects the lymph nodes. So adults and adolescents are recognized as the reservoir for transmitting this particular organism for this disease process. And then there are other species that have been associated with it, as far as Bordetella species, but those are usually just within the immunocompromised. And so over the years there have been vaccines that have been developed that help to prevent this particular infection, and so those are your Tdap vaccines, and depending on the age that vaccine is given depends on which one of those 3 vaccines that you would actually receive. It is detected through a direct detection method, so something like fluorescent staining, or where you're looking for the actual antibodies from the nasopharyngeal material, or through PCR testing. And you can also do serodiagnostics, so when you're looking some of the different tests that are looking for the antigen-antibody relationship, and so those tests can also be used to help diagnose this particular organism. If we were to look at it in a microscope, it would be very small, faintly staining coccobacilli. They can be singles or pairs. And because it is difficult to stain, it can be enhanced with a 2-minute saffron O stain counterstain. These cultures do grow at 35 degrees Celsius with humidity without carbon dioxide for up to 12 days, so they can take a little while to grow. There's no growth on blood agar or McConkey, so these organisms are plated on a Reagan-Low agar. And when they grow on this agar, they produce a small, shiny organism or colony that can resemble a mercury drop. And then over time, they can be a whitish to gray with age, and so you can see an image of that there. They are oxidase positive, catalase positive, and they are non-modal, nitrate negative, and urease negative. Francisella tularensis is carried by rodents, rabbits, beavers. Humans become infected after they've handled the carcasses or the skins of these infected animals through inhalation of the aerosols. They can also be ingested with contaminated water, so maybe one of these carcasses was in the water supply. Through insect vectors, so different there are different insects that can carry this, so getting a bite from that insect, and being bitten by one of these animals that are contaminated with this organism. And so it does cause tularemia, and the most causes of tularemia happen within South Dakota, Arkansas, Missouri, and Oklahoma. So this organism, after it has been introduced to the host, so through some sort of abrasion or skin puncture or perhaps a tick bite, then the lesion will start to develop at the site, and then it will actually start to progress to an ulcer type lesion. And so the lymph nodes that are near those lesions will become infected, and so they can become very enlarged and also become necrotic. So once the organism enters the bloodstreams, the patients become systemically ill, so they'll have like a high fever, chills, headache, aching, body aches, and then depending on how it affects the patient, it can range from very mild to mild and self-limiting type symptoms to very fatal symptoms. And so it does have different ways that it presents. So for example, glandular, which is is more common. You just have the large lymph nodes, the lymphedema. And then with this one, it's rarely fatal. And then of course you can have something like a conjunctivitis type infection. So you can see that eye where you've got those little glandular type lesions in the eye. And then it can also be something as systemic, where it causes the septicemia with that very high mortality rate. And with this one, you wouldn't have any sort of ulcers or the lymph nodes being infected. With this organism, it is non-motile, non-spore forming, strict aerobic. It does produce small, poorly staining gram-negative coccobacilli. And with this culture, it is slow growing, so it does take a couple of days to a couple of weeks to grow. And so in order to help this organism with its growth, you do want to use some sort of enhancement type media. And so it does grow better with the presence of cysteine and iron. So you would look for a media that contains those to culture this organism if it was suspected. If it grows on MacConkey, it grows very poorly, but typically it does not grow at all on MacConkey agar. With chocolate agar, it would be pinpoint, small type colonies that would require a prolonged incubation in order for it to grow. And so they could be transparent or they could be mucoid. This organism is oxidase negative, and then it can be weak or a negative catalase test, just depending on the particular strand of that genus and species. And these organisms also have a negative satellite X and V test. So if you were to culture it doing a satellite test, that's where you run that strip of Staph aureus on the blood agar plate so that you would get both X and V, then this organism would not grow. Since this organism does not gram stain very well, it is of little use to actually use a gram stain to try to help identify it. And so this organism, again, like most of these that we've discussed, are typically going to be worked up with some sort of automation or direct detection method. And so when you look for like a particular antibody stain or fluorescent antibody stain to look for this organism, or something like mass spectrophotometry or PCR testing. And so this is a biosafety concern. Laboratory employees can be exposed to this if they were to handle this organism on an open bench top. So typically when reading culture plates, the tech does look at them out in the open. So if this organism was suspected, then they would need to move those plates to the biological safety hood in order to work them up to make sure that there's no aerosols created once you remove the top from this organism.